Imagine a bustling city teeming with life. Now, shrink that city down to a size so small that you need a powerful microscope to even see it. That's the world of a cell, the fundamental unit of life. Just like a city has different departments and workers with specialized roles, a cell has compartments called organelles. These organelles work together to keep the cell alive and functioning. Cells are incredibly diverse, coming in various shapes and sizes. Some cells, like bacteria, are simple and consist of a single compartment. Others, like the cells in our bodies, are much more complex. Despite their differences, all cells share some common features. They are surrounded by a membrane that acts as a barrier, controlling what enters and exits the cell. Inside, they contain a watery fluid called cytoplasm, where many vital chemical reactions occur. The human body, for example, is made up of trillions of cells. These cells come in many different types, each with a specific function. For example, muscle cells contract to allow movement, nerve cells transmit electrical signals, and blood cells carry oxygen throughout the body. Despite their differences, all cells arise from a single fertilized egg, highlighting the remarkable ability of cells to divide and differentiate. Understanding cells is crucial for understanding life itself. By studying cells, scientists can learn about the basic processes that occur within all living organisms. This knowledge can then be used to develop new treatments for diseases, improve crop yields, and even create new forms of life in the laboratory. Within the bustling metropolis of a cell, organelles function like specialized departments, each responsible for carrying out specific tasks. These microscopic structures are the key to a cell's survival and function. Just as a city needs power plants, factories, and a transportation system, a cell relies on its organelles to generate energy, synthesize proteins, and transport molecules. Organelles can be broadly classified into two categories: membrane-bound and non-membrane-bound. Membrane-bound organelles, as their name suggests, are enclosed by a membrane similar to the cell membrane. This membrane creates a separate compartment within the cell, allowing the organelle to maintain a unique internal environment and carry out specialized functions without interference from the surrounding cytoplasm. Non-membrane-bound organelles, on the other hand, lack this enclosing membrane. They are typically composed of protein complexes or RNA molecules that perform specific tasks within the cell. While they may not have a physical barrier separating them from the cytoplasm, their unique structural organization allows them to function efficiently. The coordinated action of both membrane-bound and non-membrane-bound organelles ensures the smooth operation of the cell. Understanding the structure and function of these tiny powerhouses is essential for comprehending the complexity and elegance of life at the cellular level. In the following sections, we will delve deeper into the world of organelles, exploring their unique characteristics and roles in maintaining the intricate balance of life. Imagine a city with high walls, protecting its inhabitants and allowing them to specialize in their tasks. This is analogous to the world of membrane-bound organelles, which are like miniature organs within the cell. These organelles are enclosed in a double layer of phospholipids, forming a barrier that separates their internal environment from the surrounding cytoplasm. This separation is crucial because it allows them to maintain a specific chemical environment optimized for their particular function. One of the most important functions of membranes is to regulate what enters and exits the organelle. This is achieved through various protein channels and pumps embedded within the membrane. These proteins act like gatekeepers, selectively allowing certain molecules to pass through while keeping others out. This selective permeability is essential for maintaining the organelle's internal environment and ensuring its proper function. The membrane itself is not static, but rather a dynamic structure. It can change its shape, size, and composition in response to the cell's needs. This flexibility allows membrane-bound organelles to interact with each other and with other parts of the cell, forming a complex and interconnected network. Some of the key membrane-bound organelles include the nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, and lysosomes. Each of these organelles plays a vital role in the life of the cell, and their coordinated action ensures the smooth functioning of the entire cellular machinery. At the heart of the cell lies the nucleus, a spherical organelle that serves as the control center for all cellular activities. Encased within a double membrane known as the nuclear envelope, the nucleus houses the cell's genetic material in the form of DNA. This DNA is organized into long, thread-like structures called chromosomes, which contain the blueprints for building and maintaining the entire organism. The nucleus acts like a library, storing this vital genetic information and regulating which genes are expressed at any given time. This regulation is crucial because it determines the cell's identity and function. For instance, a muscle cell and a nerve cell contain the same DNA, but express different sets of genes, leading to their distinct characteristics. Within the nucleus, a dense region called the nucleolus plays a critical role in ribosome production. Ribosomes, as we will discuss later, are the protein synthesis machinery of the cell. The nucleolus assembles ribosomal RNA, rRNA, and combines it with proteins to form ribosomal subunits. These subunits are then transported out of the nucleus and into the cytoplasm, where they assemble into functional ribosomes. The nucleus, therefore, is not just a storage facility for genetic information, but an active and dynamic organelle that orchestrates the complex processes of gene expression and ribosome biogenesis. Its proper functioning is essential for the survival and proper development of all eukaryotic organisms. Section 5. Endoplasmic reticulum and Golgi apparatus, protein production and packaging. Beyond the nucleus lies a vast network of interconnected sacs and tubules known as the endoplasmic reticulum, or ER. This organelle plays a crucial role in the synthesis, folding, modification and transport of proteins and lipids. The ER is like a cellular factory, bustling with activity as it produces the building blocks and functional molecules essential for the cell's survival. There are two types of ER, rough ER and smooth ER. The rough ER is studded with ribosomes, giving it a bumpy appearance under the microscope. These ribosomes are the sites of protein synthesis, and the rough ER is responsible for producing proteins that will be incorporated into cell membranes, secreted outside the cell or targeted to specific organelles. The smooth ER, on the other hand, lacks ribosomes and appears smooth under the microscope. It is involved in a variety of functions including lipid synthesis, detoxification of harmful substances, and calcium storage. For example, in liver cells, the smooth ER plays a crucial role in detoxifying drugs and alcohol. Once proteins are synthesized in the rough ER, they are transported to the Golgi apparatus, another membrane-bound organelle that acts like the cell's post office. The Golgi apparatus receives proteins from the ER and further modifies, sorts, and packages them into vesicles. These vesicles act like shipping containers, transporting the proteins to their final destinations, within or outside the cell. Section 6, that's mitochondria, the powerhouses of the cell. Within the bustling city of the cell, mitochondria are the power plants responsible for generating the energy needed to fuel all cellular activities. These bean-shaped organelles are enclosed by a double membrane, with the inner membrane folded into intricate structures called cristae. This folding increases the surface area available for energy production. Mitochondria are unique organelles in that they have their own DNA, separate from the DNA found in the nucleus. This mitochondrial DNA, or mtDNA, contains genes that encode for some of the proteins involved in energy production. The presence of mtDNA suggests that mitochondria originated as free-living bacteria that were engulfed by ancestral eukaryotic cells billions of years ago. The primary function of mitochondria is to produce ATP, or adenosine triphosphate, the cell's main energy currency. This process, known as cellular respiration, involves breaking down glucose and other fuel molecules in the presence of oxygen to release energy. The energy released is used to convert ADP, or adenosine diphosphate, to ATP. ATP acts like a rechargeable battery, storing energy that can be released and used by the cell for various processes such as muscle contraction, nerve impulse transmission, and protein synthesis. Without mitochondria, cells would not be able to perform these essential functions. Section 7. Non-membrane-bound organelles, the free agents. While membrane-bound organelles operate within their walled cities, non-membrane-bound organelles are like free agents, carrying out their tasks directly in the cytoplasm. These organelles lack a surrounding membrane, allowing them to interact directly with the cellular environment and other components of the cytoplasm. Despite their lack of a membrane, non-membrane-bound organelles are highly organized structures, often composed of protein complexes or RNA molecules arranged in specific configurations. This precise organization is essential for their function and allows them to perform specific tasks with remarkable efficiency. Examples of non-membrane-bound organelles include ribosomes, centrioles, and the cytoskeleton. Ribosomes, as we discussed earlier, are responsible for protein synthesis. Centrioles play a crucial role in cell division, helping to organize the microtubules that separate chromosomes. The cytoskeleton provides structural support for the cell, helps to maintain its shape, and is involved in cell movement. Non-membrane-bound organelles, therefore, play crucial roles in various cellular processes, from protein synthesis and cell division to maintaining cell shape and facilitating movement. Their direct interaction with the cytoplasm highlights the dynamic and interconnected nature of the cellular environment. Section 8. Ribosomes, the protein synthesis machines. Ribosomes are the protein synthesis machines of the cell, responsible for translating the genetic information encoded in mRNA or messenger RNA into proteins. These tiny organelles, found in both prokaryotic and eukaryotic cells, are essential for life as we know it. Without ribosomes, cells would be unable to synthesize proteins, which are the workhorses of the cell, carrying out a vast array of functions. Ribosomes are composed of two subunits, a large subunit and a small subunit, each made up of ribosomal RNA or rRNA and proteins. These subunits come together during protein synthesis, forming a functional ribosome that can bind to mRNA and tRNA or transfer RNA molecules. The process of protein synthesis begins when mRNA, carrying the genetic code from the DNA in the nucleus, binds to the small ribosomal subunit. The ribosome then moves along the mRNA molecule, reading the genetic code 3 nucleotides at a time. Each 3 nucleotide sequence, called a codon, codes for a specific amino acid. Transfer RNA molecules, each carrying a specific amino acid, recognize and bind to the codons on the mRNA. As the ribosome moves along the mRNA, it links the amino acids together in the order specified by the codons, forming a growing polypeptide chain. This chain will eventually fold into a functional protein. Section 9. When organelles malfunction, diseases and their impact. The smooth operation of a cell relies on the harmonious interplay of its organelles. When these tiny powerhouses malfunction, the consequences can be devastating, leading to a wide range of diseases. Organelle dysfunction can arise from genetic mutations, infections, environmental toxins, or other factors that disrupt their structure or function. One example is mitochondrial disease, a group of disorders caused by mutations in mtDNA or nuclear DNA that affect mitochondrial function. These diseases can affect various tissues and organs, leading to a wide range of symptoms including muscle weakness, neurological problems, heart disease, and vision loss. Another example is lysosomal storage disease, a group of inherited metabolic disorders caused by defects in lysosomal enzymes. Lysosomes are membrane-bound organelles, responsible for breaking down cellular waste products and debris. When lysosomal enzymes are defective, these substances accumulate within the lysosomes, leading to cell damage and dysfunction. Understanding the link between organelle dysfunction and disease is crucial for developing effective treatments. Researchers are exploring various approaches, including gene therapy, enzyme replacement therapy, and pharmacological interventions that target specific organelles or pathways. The study of organelle dysfunction highlights the interconnectedness of cellular processes and the importance of maintaining the integrity of these tiny powerhouses for human health. As we delve deeper into the complexities of the cellular world, we gain a greater appreciation for the intricate mechanisms that sustain life and the devastating consequences when these mechanisms go awry.